Have you ever wondered how fluorimetry and phosphorimetry work? Ever thought about the science behind these fascinating concepts? From the vibrant hues of a chemist's lab to the precise measurements in biomedical research, these techniques play a vital role. They unravel the secrets of molecules, their luminescence, and the factors influencing their fluorescent behavior. We'll explore their instrumentation, delve into the quantitative aspects, and discuss their advantages and disadvantages. So let's dive into the world of fluorimetry and phosphorimetry, shall we? Let's start by exploring fluorimetry, a type of molecular luminescence. Now, what is molecular luminescence? It's the process where a molecule absorbs light, or photons, and then re-emits them. This re-emitted light is what we call fluorescence. At the heart of fluorimetry lie two key concepts: excitation and emission spectra. The excitation spectrum is all about the energy absorbed by a molecule, while the emission spectrum is about the energy it releases. When we measure the intensity of this released energy, we're measuring fluorescence. So, what influences this fluorescence? Several factors come into play. The environment surrounding the molecule, the wavelength of the incoming light, and the very nature of the molecule itself can all affect the fluorescence. But how do we quantify fluorescence? Well, it's a little bit tricky. The intensity of fluorescence can be influenced by the concentration of the fluorescing molecules and the power of the exciting light. So, to get an accurate measurement, we need to carefully control these variables. Now, let's talk about the tools of the trade. In a basic fluorimeter, you'll find a light source, usually a high-intensity lamp, to provide the excitation energy. This light passes through a monochromator, which selects a specific wavelength. The light then hits the sample, causing it to fluoresce. This emitted light is picked up by a detector, which measures the intensity of the fluorescence. Fluorimetry isn't just a fascinating bit of science, it's also a powerful tool with a broad range of applications. It's used in biochemistry to study cellular structures, in environmental science to trace pollutants, and even in the food industry to check the quality of our edibles. So there you have it, fluorimetry, a world of molecular luminescence where light is absorbed, transformed, and re-emitted, revealing secrets from the microscopic to the global scale. That's fluorimetry for you, a powerful tool with a range of applications. Now let's delve into spectrofluorometry, a specific application of fluorimetry. Spectrofluorometry is a type of fluorescence spectroscopy that records the intensity of fluorescence as a function of wavelength. It essentially measures the amount of light emitted by a substance after it has absorbed light, providing detailed information about the substance's molecular structure and environment. One of the key advantages of spectrofluorometry is its high sensitivity and selectivity. It can detect even the smallest concentrations of a substance, making it incredibly useful in areas like biochemistry, where it helps identify and quantify specific substances in complex biological samples. Another advantage is its ability to analyze multiple components simultaneously. This makes it a valuable tool in environmental analysis, where it is used to monitor various pollutants in water and air. However, as with any technique, spectrofluorometry has its limitations. It can be influenced by various factors, such as the pH and temperature of the sample, and the presence of other substances that may interfere with the fluorescence. Despite these challenges, the advantages of spectrofluorometry far outweigh its disadvantages. Its sensitivity, selectivity, and ability to analyze multiple components simultaneously make it a powerful tool in a wide range of fields, from biochemistry and medicine to environmental science and materials research. Spectrofluorometry, despite its limitations, is a powerful technique with a wide range of applications. Having explored fluorimetry, let's now turn our attention to phosphorimetry. Phosphorimetry is a branch of luminescence spectroscopy, distinct from fluorimetry, though they both deal with the emission of light from molecules. The key difference lies in the time taken for the emission of light. In phosphorimetry, the light is emitted after a noticeable delay, often microseconds or even seconds after the molecule has absorbed energy. This phenomenon is known as phosphorescence. Just like fluorimetry, phosphorimetry also works on the principle of excitation and emission. Energy is absorbed by a molecule, exciting it to a higher energy state. However, instead of immediately dropping back to the ground state, the molecule undergoes intersystem crossing to a triplet state. It then relaxes back to the ground state, releasing energy in the form of light, but at a slower pace. Now the question arises, how do we measure this delayed light emission? That's where the instrumentation comes in. The core components include a light source to excite the molecule, a delay mechanism to pause between the excitation and emission measurements, and a detector to measure the emitted light. The delay mechanism is what truly sets phosphorimetry apart. It allows for the measurement of phosphorescence even in the presence of more immediate fluorescence, expanding the range of molecules and environments we can examine. That's a brief rundown of phosphorimetry, another intriguing aspect of molecular luminescence. But what makes phosphorimetry so important, you ask? Well, it's all about the advantages this technique offers and the wide range of applications it has in various fields. Firstly, phosphorimetry is highly sensitive. It allows us to detect even very low concentrations of a substance, making it an extremely powerful tool for analysis. This heightened sensitivity is due to the fact that phosphorescence occurs in a dark environment, free from the interference of other luminescent sources. Another advantage is its specificity. Phosphorimetry can distinguish between different substances based on their unique phosphorescence characteristics. This specificity can be further enhanced by manipulating the temperature and the timing of the measurement. Now, no technique is without its drawbacks, and phosphorimetry is no exception. One of its limitations is that not all substances phosphoresce. Only those with long-lived excited states can be analyzed using this method. Moreover, the technique requires a relatively long measurement time due to the slow decay of phosphorescence, which might not be suitable for all applications. Despite these limitations, the applications of phosphorimetry are vast and varied. In the field of analytical chemistry, it's used to identify and quantify trace amounts of substances. It's particularly useful in environmental monitoring, where it can detect pollutants in air, water, and soil samples. In the pharmaceutical industry, phosphorimetry aids in the quality control of drugs by detecting impurities. It's also used in forensic science for the detection of certain substances in criminal investigations. Furthermore, phosphorimetry has found its place in the field of material science. It's used to study the properties of materials and to develop new ones with desired characteristics. For instance, the technique has been used to develop phosphorescent materials for organic light-emitting diodes, or OLEDs, which are used in displays of various electronic devices. Despite its limitations, phosphorimetry is a critical tool in many scientific and industrial applications. So, whether it's ensuring the safety of our environment, validating the purity of our medicines, solving criminal cases, or even enhancing our viewing experience on electronic devices, phosphorimetry is indeed playing a significant role. That was a quick tour through the fascinating world of fluorimetry and phosphorimetry. We've journeyed together through the intricacies of these two compelling spectroscopic techniques, each with its own unique characteristics and applications. In our exploration of fluorimetry, we delved into the captivating phenomena of excitation and emission spectra, along with the quantum world of molecular luminescence. We've seen how the measurement of fluorescence can be impacted by various factors and how this knowledge can be harnessed for quantitative analysis. We then ventured into the realm of spectrofluorometry, taking note of its substantial advantages. The increased sensitivity, specificity, and the ability to analyze multiple components in a single sample are just a few of the highlights that make it an essential tool in many scientific fields. Transitioning to phosphorimetry, we've unveiled the distinctive instrumentation involved, its advantages, drawbacks, and broad range of applications. The ability of this technique to detect minute quantities of substances is truly remarkable, making it indispensable in various areas of research and analysis. Remember, the world of spectroscopy is vast and ever-evolving. These techniques are more than just scientific concepts. They are tools that help us understand and interact with the world around us on a molecular level. So stay curious, keep exploring, and remember to subscribe for more because you won't find me later. See you in the next video.